Victor Hruby. His company is MCR Therapeutics. Victor?
Hi, thanks. Thanks for sticking around. Full room. Like to see that. So I want to tell you about a new company that Minnie and Kai started about 6 months ago or so. And my reasons were quite different than hers. I wanted to start the company out of frustration. I've been working in the melanocortin area for 45 years and made many of the discoveries that have been made in this field with my biological friends and some of my chemical friends. And there's only one drug on the market. It's something we discovered 35 years ago. So what's going on? The MCR system is one of the primordial systems in animal life. So it's involved in virtually all of the critical biological functions that keep you alive every day. And I'll say a little bit about that when I get into my talk. But MT1 and MT2, two of the compounds we discovered, are in clinical. One of them is already on the market, the other one's in clinical trials, but you can also get them on the internet. And I think it's a much larger business on the internet than it is. So I'm kind of frustrated. Why is this that something we discovered is a bigger market on the internet by millions and millions, probably even $1 billion, I don't know. It's a huge market anyway. Okay, so let's see. How do we move forward in this thing? Maybe I turned it upside down like everybody else. Okay, let's see if that works. Yes, how about that? Okay, so where we are right now, that led me to think we could get this started right now. If we have preclinical studies for many MCR selective ligands, I'll tell you what these are in just a little while. We have been examining multiple delivery systems for these peptides and peptidomimetics. We have already made applications to the FDA, some of them 25 years ago, but we are ready to make FDA clinical trial applications very soon, and we hope to have clinical trials in 3 to 5 years, and more recently, maybe even much sooner, because we have we are having some discussions with people in China that might allow us to start much sooner. Okay, so what is the melanocortin system? Initially, it was 2 receptors that were known for biological reasons. All of these receptors were cloned in the 1990s, and when they were cloned, 3 additional receptors were found, the MC3, 4, and 5. These are involved, as I said, in most of our necessary functions, from pigmentation to pain to stress, fear, flight, obesity, energy homeostasis, pain, sexual behavior, learning behavior, immune response, on and on I can go. So this system is critical for almost all of our major degenerative diseases. Okay, here's the peptides for this system from another primordial gene called POMC. Now POMC is very interesting in that it contains these 3 peptides, alpha MSH, beta MSH, and gamma MSH, all of which have this key core sequence. Most of the work's been done with alpha MSH. Then it contains ACTH, which also contains this core sequence, which has been held up through all of these 700 million to 1 billion years of evolution. It's very interesting. These are non-selective ligands, very non-selective for the 5 receptors, except that alpha, beta, and gamma MSH do not interact with the melanocortin 2 receptor. Okay, so what are these receptors doing? Now, the other reason why we think that we can go on in this system is that there are lots of new kinds of academic and biotech studies that amplify compelling MCR target development opportunities. Now, one of these is called biosignaling now. Now, this is actually nothing new. It's just a new name for something that's known for a long time, that is, for many hormones and neurotransmitters, there are alternate ways in which you can transduce the message. And this became called biosignaling more recently, but I always wonder, why do we change names for something that's been known for years and years? It's very interesting. And it's a really hot topic because a lot of young people never heard of, you know, of what happened 25 years ago or so. Anyway, it's very interesting, but it's very important because this gets people now to see things from a new perspective, and that always helps, right? Okay, then there's a lot of recent preclinical trials to study some of these newer things like neurodegenerative diseases, stress, cancer, pain, IBS. And then here's the clinical trials. Okay, Clinovil, which is MT1, Palatin, which is MT2 with the carboxylate terminal, and then another compound associated with these. Again, this is all first round structure activity from our perspective. Anyways, but they're in clinical trials, and one of them's out in the market now. We also have now developed agonists, antagonists, and inverse agonists that are highly selective for all four of the receptors we're interested in, which is MC1, 3, 4, and 5. And we also have inverse agonists for those that might be interested. We don't yet have a medical application for an inverse agonist, but we have them. We have both orthosteric and allosteric, to use the modern terminization for this stuff. But of course, we already knew that there were orthosteric ligands 25 years ago. And then we have peptides, peptide mimetics, nonpeptides, potent, selective, and bioavailable. Okay, in design, I'm not going to say very much except for a couple of things that make our situation now very unique, at least to my knowledge. And, wrong way. Which way? And that is that we now can design our molecules based on receptor selectivity. Now, this took us about 12 years to develop in collaboration with others, where we did both domain swapping and site-specific mutagenesis to show what is the differences in the binding site for the MC1, 2, 3, 4, and 5 receptors. Now, we've tried to get support for this, but of course people say you can't do this because there's no X-ray crystal structure. Well, X-ray crystal structure is just a picture. That's not the way biology works. The way biology works is that you have a function. So we did this with function. And that's better than X-ray crystallography because we now have knowledge of these receptors so we can design specifically for each of these receptors with this model. So, you know, X-ray crystal structures are nice, but they're just a picture. We've also developed a lot of pharmacological and biochemical tools. I list some of them here, and we have many others that are being developed or have been developed already by ourselves or our collaborators. By the way, we collaborate very extensively with people in the medical school and biology departments in this area for many years. So I'm just going to talk about a few applications. One is for melanoma prevention. This was our initial goal 25 years ago when we did our clinical trials with MT1. And in its infinite wisdom, Johnson & Johnson decided to drop this. Big mistake, but it'd probably be $1 billion drug now, but that's what happened. So it's on the, you can get on the internet. Johnson & Johnson doesn't sell it, but the internet does. But it's very important because melanoma cancer is one of the fastest growing cancers. It's also, there are no good treatments. And yet, we think we have very good ways to both detect it very early on, to treat it, and to prevent it. So the first thing here has to do with prevention. And here's basically what it means is, instead of going to become tan, as you may know, a lot of people, light-skinned persons, are very susceptible to melanoma, especially with red hair. If you are a black person or an Asian person, your chances of getting melanoma are about 30 to 50 times less than those of a white person. It's the fastest growing cancer in the world because more and more people are spending their time vacationing in the sun. There's a big business called salons. Multi-billion-dollar business for people for tanning. But you're exposed to UV radiation. This tans you without UV radiation. what does tanning do? Why does it prevent cancer? Because the melanin which is formed, if you look at it, comes and sits right over the nucleus of the cell. Not just over the cell, but over the nucleus of the cell. So if you want to prevent melanoma cancer, by the way, we have shown, and published this quite a few years ago, that it protects against 95% of the UV radiation that hits the nucleus. Okay, so this looks like so we think it's very promising. And so we've been evaluating peptides for these. There's some other new applications too that I mentioned before. MC4 selective agonists for neurodegenerative diseases such as Alzheimer's. This is a preventive again. And maybe a treatment too, but I won't get into this. There's not enough time here. We want to explore the MC5R. This has been around for a long time. We know very little about it except for sebaceous gland secretion, but it seems that it's also very good for treating stress. And so we're looking into different kinds of stress situations in animals and trying to find out where this receptor is involved in stress. And then finally for the MC1R selective is also for the treatment of melanoma cancer. Now this is a surprise to some people, especially the medical profession, which has always claimed that actually melanocyte stimulating hormone stimulates melanoma cancer cells. This is not true if you have a selective MC1R receptive ligand. And we published on this 20-some years ago. But medical doctors don't read the literature, and maybe most of my scientific colleagues don't either, because people are still saying this. So, so I just want to say that MC1R selective agonists actually kill cancer cells, melanoma cancer cells. Not all melanoma cancer cells, but the vast majority that we've tested so far. Okay, so here's just to give you, we really have real structures. I'm showing a few of them. We have many, many patents in this area and so forth, and I'm not able to show you some of the structures because we still are in the process of patenting them. But here's one of the patents which we just recently got, U of A 17-016, which is for neurodegenerative diseases, and we're working with people in the neurosciences to study its effect on neural regeneration and neurodegeneration with aging, with age. So since Alzheimer's is a disease of aging, we want to find out how this system affects the neurons that are associated with learning and behavior. Here's another one of our MC1R selective ligands for pain and for circulation. So it turns out we had the first people that had an MC1R selective antagonist, and we were able to publish with our colleagues in Canada that this pain, melanocortin receptor also affects pain, but only in females. So it's a very, very interesting situation. There's a difference between the pain system in males and females. I think we already knew this because of childbirth, and it has not been demonstrated, however, that this is related to childbirth, but it may be. And so we're looking into this as it's a very interesting situation. There's also other possibilities here that have come up in our collaborative efforts with some of our colleagues around the world. So this is its effect on cholesterol transport in macrophages, effect on endoavailability and endothelial dysfunction, and on hyperalgesia. So there's lots of other possibilities here where this might be very important. So in conclusion, and by God, I stayed on time. How can that be? Hruby never stays on time. Anyway, we think we have potential applications for a wide variety of melanotropin ligands to health and disease. Several of our peptides and peptides are already in clinical trials. By the way, I didn't mention a few of these. Other companies have come in and taken a few of these. But for the most part, these things are still available. So MC1R ligands are now well developed and ready for clinical trials for tanning, pigmentation without sun, replacing salons and preventing skin cancer, multimodal NMR ligands. We have multimodal methods now for converting these highly selective ligands so we can use different imaging methods with one ligand. So this is going to be we think, very useful for cancer detection, especially melanoma cancer, and then for treatment of cancer for reasons I told you earlier. So there also are excellent ligands available for sexual function, feeding behavior, neurodegenerative diseases, stress, and inflammation. And with that, here's the people in my group and a few of our collaborators that have done the most recent work. I mean, I've been involved with this for many years. We have over 500 publications in this area, so obviously I work with lots of people all over in my lab, first of all, for many years, and with collaborators at the University of Arizona and throughout the world for many years. And I want to thank them, because I wouldn't be able to be here talking about this new company without their work. Thank you.